For today's discussion, I have the absolute pleasure of introducing Antoine Remiot and Alexis Davin. Antoine is one of our more tenured experts, and he heads Enside's solution engineering team. Alexis is part of Antoine's team and is one of our brightest clinical supply optimization experts. I'm Dan Caramore, one of our US account executives, and I'm your moderator for today's webinar. So for those of you that do not know Enside or are just learning about Enside, for the past 20 years, we've worked with pharma and biotech companies delivering improved efficiency, visibility, waste savings, and a faster time to market for over 12,000 clinical trials. Through our team of experts and risk-based optimization software, Enside has successfully provided the most accurate forecasts and optimized recommendations for clinical trial supply activities, while always making sure that our patients and the patients we're working with receive their dose. With that said, and my brief introduction, it's my absolute pleasure to introduce Antoine. So I'll pass it over to him now, and I will talk with you all soon.
Thank you, Dan. Thanks a lot. So yeah, for today, what do we have? So first, we're going to focus on the influence that trial design can have on clinical supply chain, because there are a ton of overlooked parameters that have a major influence on waste, on cost, on supply chain risk. So we're going to discuss that first. Then we're going to illustrate what we just discussed with a couple of case studies on protocol design, but also on supply chain design. And then finally, we'll conclude this presentation with some tips and tricks on how to make your trial design more supply friendly. What are the parameters that you can influence to reduce waste, to reduce costs, but also how to change the organization, how to implement processes to make sure that this becomes feasible, because this is not always a given in all pharma organization. So we're going to give you some tips and tricks. So without further ado, let's start. And here on the slide, what you can see is a typical clinical supply chain process. So we start on the left side with everything related to manufacturing, drug substance, drug product, and then on the right side we have the trial supply chain with the central depot, the different local depots, and the sites. And I think we can all agree that our industry is pretty wasteful, because we tend to have a lot of waste that is not accounted, that is not really tracked. But these days, reducing waste has become one of the primary focus on the industry, because we could do so much more if we were to reduce waste. And we've observed that on the manufacturing process, up to 20 to 40% of that waste could be reduced. What do I mean by drug waste? I'm talking, for example, when you have to produce a minimum batch size of, 5 liters, for example, and then you convert everything into DP, even if you don't have a specific demand for that DP. You convert it anyway, and then it's just waste. On the trial supply chain aspect, we also have a ton of waste. Kits expiring at the site level. Sometimes clinical supply chain manager, they plan for an average of, 150%, while they might only need 50%. So that's also pretty wasteful. And here, we believe that up to 60% of that IMP waste could be easily avoided. So I'm not saying, don't get me wrong, I'm not saying that all waste should be avoided. There is a necessary waste in our industry, because you need some waste to cover the uncertainties linked to randomization, to the up-titrations, to the dropouts. So there is definitely a necessary waste. What we want to tackle is the unnecessary waste, the extra buffers that we take just in case. That's exactly what we want to tackle. The bad news is that these days there are a couple of trends that are not going in the right direction, not going in the direction of reducing waste. First of all, the complexity of trials is increasing. And by that, I mean the adaptive trials are becoming more and more prevalent. We have complex titration schemes, different cohorts, overall complex designs that are really increasing the complexity of the trials, so that, of course, it becomes more complex to manage and more complex to lower the waste in such trials. On top of that, regulations are not helping us. We've seen that approvals are taking a lot of time. Sometimes we have new regulation in Europe, in China. We need packaging and labeling on primary and secondary packaging. So that also reduces the possibility to extend the expiries. So that further increases the waste. Drugs are becoming more and more complex to manufacture. So that reduces the usability periods, that reduces the shelf lives. And of course, if you have short shelf lives, you increases your waste. And overall, as you all know, pharma organizations have been able to produce a new drug in only a matter of months. It was incredible, but now it tends to become the new norm, unfortunately for us. So we expect faster timelines. So that means that we don't have as much time as before to plan in a smooth way or IMP or clinical supply chain. So a couple of months ago, we did an analysis at Inside, and analysis over hundreds of trials over multiple indications from phase 1 to phase 4. And we tried to identify the main drivers of waste. Where does the waste come from? And I know each trial is different and each indication has its own challenge, but we came up with 6 different categories that are really driving the waste. The first one is linked to unexpected events. So delays in the recruitment, Brexit, COVID, dose levels, titrations that are not ending at the dose level that you were expecting. So all those unexpected events are generating some waste. Then we have everything related to planning. How do you plan your quantities? Do you use an Excel sheet? If so, what kind of overage do you apply? 100%, 150? It depends. So the forecasting and planning aspects is obviously generating a lot of waste. Then we have everything related to the site resupply strategy. So how often will you resupply your sites? What will be your buffer levels at the sites? Then we have the lack of monitoring. So I talked about the unexpected events. So that's one thing. But if you don't react in time, then it will get even worse. So if you don't have monitoring capabilities to react to a delay in the recruitment, for example, then you will pack and label as many quantities as planned before without having observed that the recruitment is not on track. So you need a flexible supply chain, flexible strategy to be able to react to those unexpected events that will anyway happen. It never unfolds as planned. Then we have the supply chain design. What are the shipping lead times? How many depots do you have? How often do you pack? And then the trial design, everything related to the protocol, to the kit design, and so on. So we have a small poll for you. Dan, you want to take over?
Yeah, thanks, Antoine. Appreciate it. So we've given you some main drivers of inefficiencies in the clinical supply chain. We'd love to hear from you here. If you navigate to the right side of your screen, you should see in just a few moments a new poll populating. So our question is for the chat, according to you, what's the most impactful driver of waste? Out of these 6 options, we've bundled the last 2, supply chain design and trial design, into one. But please, and again, anonymously answer in the poll on the right-hand side of your screen. And Anton, we'll just give it a few seconds here as the answers start to go in.
Yeah, we see a lot of answers. That's very interesting. And obviously, you may have different answers based on the type of trial that you use to work on. If you're working on rare diseases, you're not going to have the same answer as if you were working in oncology or in diabetes, obviously. Okay, so unexpected events is received quite a lot of answers, as well as supply chain and trial design. Don't have a lot of love for planning nor for the lack of monitoring, but the site resupply strategy also received quite a lot of answers. So everything related to the IRT parameters, that's interesting. All right, so the top 2 answers are unexpected events and supply chain design and trial design. And from our experience, this is the pie chart with the output of the analysis that we made. And what we can observe is that The supply chain design and trial design represents almost half of the waste generator factors, in a sense that that's really where you can impact significantly your future waste. So if you manage to optimize trial design and supply chain design before the first patient in, obviously we don't want to do protocol amendment, we want to have something supply friendly from the get-go, then you can really significantly reduce waste. And I will give the floor to Alexis, who's going to talk more in details about trial design and supply chain design.
Hello, everyone, and thank you, Antoine. So indeed, as Antoine just mentioned, the trial design and the supply chain design are two main sources of waste in clinical trials. And if we first start to focus on trial design, as you can see on the slide, we have made a list of decisions that are taken at trial design phase. And in fact, most of the time, the waste due to trial design comes from a misunderstanding of how important these decisions are for the waste. Indeed, the visits window, the visit schedule, the kit design, the different titrations, the screening period, or the kits directly shipped to the patient's house, the country selection, or the stratifications of the different patients, all these decisions might be considered as a small detail, but at the end of the day, they can have huge consequences on the waste. The second point is the supply chain design. And as I like to say, if we were able to manufacture and to ship the kits instantly, we would have no risk of shortage and we would have no waste in our trial. Unluckily, the reality is a bit more complex than that because the supply team has to deal with a lot of constraints. For instance, the manufacturing frequency, the sourcing strategy, the potential bottlenecks at packaging. For instance, some CMOs can only produce by batches of 1,000 kits, and many others. And the point is, in order to avoid to increase massively the waste, the supply team has to take into account all these constraints together. And from time to time, a small detail can increase massively the waste. For instance, in a clinical trial with weekly visits, if you cannot ship in 5 days to the site, but instead you can only ship in 8 days, well, you will increase the waste because you will potentially need to cover with your buffers not only one visit, but maybe 2 visits. And we will see a couple of examples right now with some case studies to show you how important it is. So indeed, now it's maybe the good time to share with you some examples where we were inside with clients, tried to challenge the trial design or the supply chain design. So as Antoine mentioned, for the sake of this webinar, all the case studies that are then real trials were completely blinded. And here we can maybe start with the first example where we challenged the protocol design. So in here, we challenged the minimum screening period. So just to give a little bit of context, so we are in a classic trial where the patient comes at site, gets the screening visit, and the patient can be randomized 1 day after the screening visit to get either active or placebo at site. Of course, in 1 day, we cannot ship anything on site. So to ensure the dispensing of the patient, 1 buffer of active and 1 buffer of placebo need to be on the site when the patient comes. When we looked at that trial with the supply team, we simulate and we got the 2 KPIs that you can see just below. But as there were a lot of sites to be open for a very small target of patients, we wanted to suggest to increase the minimum screening period up to 10 days. Why? Because this would give enough time to send the buffers at screening rather than at site initiation. And what we see is this brings a 14% cost reduction, because with this strategy, we avoid to replace the buffers again and again, even if no patients shows up. And the point is that this is always a pretty good, let's say, standard to try to have a long minimum screening period, as long as it doesn't affect the patient friendliness. The second example, we are still challenging the protocol design, and here we challenge the visit schedule. Especially, we tried to modify the visit interval. So to give you the feature of the study we're working on, in this study, the IMP had 8 months of shelf life, which is not a lot. The dispensing was 3 kits every 3 months, and we were dealing with long lead times countries like Brazil or Turkey, with over 60 days of lead time. So all these constraints together led us to a usability period of only 3 months at site level. The problem is that the waste, as you can see in the baseline, the waste was over 200%. So the overage was over 20%. So we tried to reduce the overage. And for that, as we couldn't change anything for the shelf life, neither for the country selection, we tried 2 scenarios. One, changing the dispensing to 2 kits every 2 months, and the other one to 1 kit every month. That, as you can see, can bring up to 25% and to 45% of cost reduction, which is massive. The problem is that asking patients to come back to the site if they're living very far away is not very patient-centric. So of course, in terms of cost, it's great. But when we discussed with the clinical team, that concern was raised. But at the end of the day, what was implemented was 2 kits every 2 months, because even if the patient had to come back more often, looking at the trial and at the drugs the patient had to take, the clinical team found it was not a bad idea to also try to provide a better follow-up to the patients. So here it is typically the kind of situation that we really like, because it's a win-win situation for the supply and the clinical team without affecting the patient centricity. Here is a last example where we challenged the protocol design. Again, we challenged the visit schedule, but this time we tried to add a visit. So maybe to give a bit of context before explaining the suggestion we made, in this study, a patient had first a beginning of treatment with a small visit interval. So the patient came back every 2 weeks, then every 4 weeks. And then at week 12, the visit interval was 8 weeks. And at week 12, on top of that, there was a possibility to titrate, so to have a dose change. And as this dose change was a new kit type, well, we needed then to put as a buffer a high number of kit types, because we needed to last 8 weeks. And for the waste, it was really a terrible thing. So our suggestion was, instead of letting the treatment like this, maybe we could try to add a visit, what we called a control visit, on 2 weeks after the week 12. So in this case, when the patient came, they would receive only 2 kits. So we decreased then the level of buffer from 6 to 2. And they came back, they should just come back 2 weeks after to get the rest of the treatment. And what we see as an impact was a 20% of cost reduction. And on top of that, after this had been implemented, what we saw is that the assumption for the dropout rate around the week 12 was much lower than expected. And the reason is that the patient, after changing dose, could have potentially side effects. And if they were coming 8 weeks later, maybe they would have suffered too much from their treatment, so they would maybe have dropped. While here, after 2 weeks, there was always a follow-up, a closer follow-up to the patient, and so they stayed longer and they stayed more in the study. Now I will let the floor to Antoine to explain you some case studies related to supply chain design.
Yep. Thank you, Alexis. So yeah, here we discussed protocol design and how it can impact waste, how it can impact patient centricity. That's very important. Here, let's take a moment to talk about supply chain design. And what do I mean by supply chain design? I mean the distribution network that's that you're working with. How many local depots do you have? What are the shipping lead times? How often will you pack? Will you source? And here, in this example, on the right side of the screen, what you can see is a screenshot from our optimization software. The blue dots represents depots. So we have a local depot, a central depot in Europe. Then we have a couple of local depots in Brazil, Russia, and so on. And then the orange dots represents the sites. And right below, you can see the shipping lead times to reach those different locations. What is interesting is that we often consider the waste as being one single KPI for a trial. We have a trial with 60% of waste, 50%, 30%. But we never think about the waste as a KPI per location. And here, we did the exercise. We computed the waste per country. And what we observed was quite interesting in the sense that the waste was very different from one country to another. As you can see, in countries directly distributed by the central depot, such as France, Spain, European countries, we have a very low level of waste, 21%. On the other extreme, for countries with a local depot with long lead times, we see a very high level of waste, 70, 80%. Why is that so? It's explained mainly by two factors. The first one is that if you have a limited shelf life, the time that is being spent during shipments is reducing the usability period of the drug. So if you have, a shelf life of 6 months, if you already spend 2 months in transit, then you only have 4 months to use the kit. And I'm not even talking about the do not dispense dates. On top of that, the second factor is that as you have long shipping lead times, you have to send a lot of kits, because if it takes 2-3 months to send new kits, you don't have time to react. You cannot react quickly. So you have to send enough kits to the local depot to build a big safety stock to cover anything that could happen. So to cover any kind of uncertainty, you need to send enough kits. So that's why in countries such as Brazil, such as Russia, we tend to see those big overages. What we often like to do as well is to compute the waste per country, but also the cost per patient per country. And then we have interesting discussion with Clinops to see, okay, if you want to add 30 patients, where do we put those patients? I know that supply is not the deciding factor, but it's an interesting piece of data to consider, because if it's cheaper to recruit 50 patients in Europe than 12 in Brazil, maybe it's worth discussing to see what's the best option. A second thing that could be done here is to try to merge local depots together. For example, here we have a depot in South Korea, another one in Australia. What if we could have a single hub in Singapore supplying both countries? We would have a single safety stocks rather than 2 safety stocks. That would further reduce the waste. Then something that is also extremely important is the release frequency. How often will you pack and label? How often will you source? Well, obviously, it really depends. The answer depends on the type of trial that you're working on. It depends on the shelf life, depends on the visit interval. You will not pack at the same frequency if you have weekly visits or 1 visit every 3 months. Depends on the shipping lead times, study duration. Sometimes it's worth packing a single time at the beginning of the trial and be done with it. Sometimes it's important to have more frequent releases. So here, on the small graph that you can see on the bottom left, you can see the concept of overlap. So if we have our first campaign in pink, you have the first release, the expiry, then you have second release, the expiry, and then we have the overlap. The longer the overlap, the smoother will the expiry replacement events be. in the sense that we will have more time to replace smoothly the buffers at the site and at the depot. On the other hand, if you have a short overlap, you won't have a lot of time to replace those buffers, and you will generate more waste. So in this specific example, we were dealing with a very expensive trial, close to 100 million of supply costs. And why is that? Is because it was a large phase 3, multiple hundreds of patients. And on top of that, we're dealing with an expensive comparators with a short shelf life, only 8 months of shelf life. And we tested different scenarios in our software. What if we would pack every 2 months? What if we would pack every 4 months? So you see the overlap is now reduced, and we see that the supply cost, the supply budget increased by 35%. The overage is obviously increasing. We're still dispensing the same amount of patients, but we need more kits and fresher kits, so we need just higher quantities. And finally, we tested what if we would source only twice a year, every 6 months. Here we see that the supply cost and the overage goes up, but on top of that, we start to have patients missing their doses. So that's absolutely unacceptable. We absolutely want to avoid that, but there is a good reason for why did patients start missing their dose. Because with such a short overlap of 2 months, it would be shorter than the shipping lead times to reach some of those countries, such as Brazil. So obviously you have some risk, and that's something that you want to test early on, before deciding on the release frequency. Because if you don't test, if you decide to source every 6 months, then you will have patients missing the dose, and that's really something you don't want. So we want to avoid risk. We want to test different scenarios. We want to involve Clinops early on to ensure patient centricity and to ensure no supply chain risk, such as drug shortages or patient missing their doses. So now let's take a step back and try to see, okay, this looks good, this looks very interesting, but it does not apply to my organization. We don't have the processes in place. I don't know where to start. I fully agree that in a lot of organizations, the supply chain department is not able to influence those decisions, because it's patient centricity is at the heart of every decision, and it should stay that way. I fully agree. We received a very interesting question that even the protocols are designed to meet scientific needs, even if the design creates some waste. I don't see that that's something that can be easily modified. Agreed, because the primary focus should be indeed the scientific needs. But what if we could make some modification that would not impact the patients? Earlier, we saw in one case study, what if we would change the minimum screening period from 1 to 10 days? It's only feasible, obviously, if the clinical team accepts to do so. If we see that there is a need for a biopsy, it always takes a couple of days, it would not impact patients at all, then okay, let's do it because it reduces weight without impacting patients. Sometimes something that we also do is just to make trials feasible. Because if you're dealing with short shelf lives, with budget constraints, if you don't apply any kind of optimization, the trial would just not be initiated, would not start. So we try to tweak some parameters to make it happen, just simply to make it happen. Otherwise, the trial would just never start. And again, without the technology to test those different scenarios, to assess the impact of changing those parameters, it's impossible to get the right KPIs, to have the measure of the cost, the measure of the risk. So it's important to be well equipped. And my first recommendation is indeed to move away from Excel. Why is that? Is because in Excel, we tend to see that clinical supply chain managers rely on a conservative demand. Why is that? It's because when you have to deal with weight distribution, patient up and down titrations, different cohorts, reconciling the actuals, dealing with Excel can be very complex. Expiry events, so that's also something complex to handle. And the overage is most of the time an educated guess. It's not like something the system tells you. It's, I believe we should have 40%. I believe we should have 60%. Sometimes it's too much. Sometimes it's not enough. And then you have to deal with some risk of drug shortages That's absolutely something we want to avoid. What we want to suggest is to rely on a purpose-built forecasting tool that gives you a comprehensive view of your supply chain strategy, gives you KPIs on cost, on waste, but also on patient risk. Again, it's not worth reducing the cost if it's being done at the expense of the patient. That, I think we can all agree on that. We don't want to miss a single patient. But if we can optimize costs, optimize waste without impacting patients, then let's do it. I don't see why we shouldn't do it. Such a tool promotes really the collaboration between the different departments, between clinical supply chain team, the clinical supply chain team, and clinical operation, of course, but also with your CMO, your IRT, your CRO. So you need some data to be able to discuss, otherwise it's just best guesses. Yeah, go ahead.
So designing your trials in a more supply-friendly way is not something easy. And as Antoine mentioned, the first step is to try to move away from Excel. Here, let's try to discuss of a couple of good practices that we have identified and inside that are always good to put in place if you want to make your trials more supply-friendly. So as we already seen earlier, having a minimum screening period that is longer than the maximum shipping lead time, excuse me, than the maximum shipping lead time is always key, because thanks to this, you can test if it is better to send your kits at the first patient screened, and you avoid, in this case, to replace all the time the kits, even if no patients are showing up. So that's a first good practice. A second one is to make sure that your kit design is aligned with the visit interval and the dose levels. So for instance, if you're in a trial where you have multiple dose levels, like 50 milligram, 100 milligram, 150 milligrams, you can either have different kit types. This means then different kit types, so more buffers on site, so increasing the waste. Or you can also try to test, if it doesn't affect the patient centricity, to maybe use combination of 50 milligrams of active and placebo. And combination of that, you can make all the different dose levels, and this might then decrease massively your waste. A third pillar is for the IRT. So as Antoine mentioned earlier, all the clinical trials are different. We all know that. But as the clinical trial are different, so the IRTs are as well. They all have different capabilities and resupply algorithm. And you need to make sure that the IRT that you will choose is the right one for your trial, and that you can customize the parameters to match with your trial. So for instance, if you have the big difference of lead times between your countries, make sure that you have an IRT that has the capability to put different resupply window depending on the country. Same for the kit type. If the different kit types are not following the same visit schedule, make sure that the IRT you will choose for your trial has the capability to have different do not dispense per kit type. These are small details maybe, but that can, at the end of the day, make a big impact on the supply waste. A fourth pillar is when you know that you will have a data readout milestone, when you know the date, try to put the releases after if you can, because it's always risky. You never know. Your trial can be on pause for a moment. Your trial could be canceled, and if it is the case, all the packages that you have released before, you won't be able to use it, and so you're losing budget. So, of course, it's better if nothing is canceled or nothing is paused, but in any way, it's always a good practice to apply. And the last one might be a bit basic, but the bottom line is try to have an efficient web network. So it means countries with low lead time. Try also to have the minimum amount of local hubs. because you can really see this as every time you add a hub, it means adding a step between the manufacturing and the patient dispensing, so increasing the waste. Of course, we know here these are good practices to try to put in place, but basically the goal is to try to have your trial more supply friendly. And we know that it's not easy to get there. It won't be done in one step, because we were speaking about good practices, but also about a new way of working, a new organization. And for that, we would like to show you some processes that you could put in place to try to reach this. And the first one is when you receive a trial under a draft, it's extremely important to try to test all the potential scenarios on that could maybe have an interesting output in terms of supply. So the goal, and this is a good segue to the second point because they're linked, the goal is to try to train yourself, to train the supply team to identify how to reduce the drug waste, how to avoid shortages. And thanks to this, if you can spot all the different scenarios that are interesting to do, you can then simulate them, have real numbers to show, and then you can start just keeping the more interesting one and build case studies on this. Because thanks to that, with your case studies, these will be a very good support for the discussion with the clinical team, with all the different stakeholders. And the goal here is really try to enhance the collaboration between the team to get the best win-win situation. A fourth point is, and as we mentioned already earlier, so if the supply can be involved in the selection of a vendor, it's always great if it impacts the supply. So we discussed about the IoT, as they're very different. It's important to customize it and to choose the right one depending on the parameters. But it is the same for the CMO. They have also, all the CMOs are not exactly the same. They have typical way of working and typical constraints. So let's make sure that they don't have constraints that are extremely, complex for the trial, for the supply to handle. So how many days of lead times do they have to reach a certain regional depots? How many regional depots do they have? So all these questions, it's always great to ask them and to make sure that you select the right vendor. And the last point, when we discuss with the industry, we understand that not a lot of companies are making or performing retrospective analysis. And the point is that this can be dangerous because the retrospective analysis is the best way to just follow your waste. Because the waste is not only a KPI that we define at the beginning of the trial, it is something that we should follow along the trial. And the good thing with the retrospective analysis is to be able to check all the lessons learned of this trial, to try to apply and to find solutions for the next time the problem might arise, but also to try to locate, for instance, the waste. For instance, in this example, we see that we had 46% of dispensation rate, which means more than 100% of overage. And what we see is that the biggest part of the overage is located at site level. So now this is done, but for the next time, for a trial that has a design pretty similar to this one, well, what could we do to make it better, to try to decrease this waste? So it is maybe decreasing the buffer levels, maybe they were too high. Maybe it is trying to see if the titrations possibility were not too complex, and so we needed to have a lot of buffers to cover this. Maybe we can simplify, or maybe our assumptions of dropout rate were just not accurate. And there are many others. But the goal with this retrospective analysis is, of course, not to pinpoint any mistake, any problem. The point is just to try to learn from this and do better for the next clinical trial.
Thank you, Alexis. Thanks. So yeah, we've seen how to improve trial design in terms of supply chain efficiency, how to improve supply chain design. And here, I just want to show you a small graph that shows the level of waste in our industry. So this is a graph that, this is an analysis that has been performed over 1,000 trials across, again, different indications, different phases. We've observed that without any kind of optimization, the average IMP waste level was around 70%. That's incredibly high. You may believe this is not the case in your organization. I sure hope so. But this is what we've observed. And why is that? Is because people don't have the same definition of overage. Some people just consider that the overage is what they add on top of the demand plus the buffer plus the safety stocks. But buffers and safety stock also expires and are also part of that waste. They never consider the waste in terms of direct product, in terms of direct substance. So overall, 70% of waste without any kind of forecasting tool, without optimization. And what we see is that the earlier we optimize, the lower this waste level will be. And this level of 22% is something we're very proud of that we've been able to achieve. If we optimize trials early on during the protocol writing phase, then we can get down to 22%. This is not something that is implementable in like a day or a week. This is something that we've been working on with our partners for a long time. It's now being part of their standard process. Every single new trial goes through this optimization phase of optimizing parameters, optimizing protocol design, involving the clinical supply chain in the IRT selection, in the CMO selection. And that's how we are able to reach those low level of waste. So overall, again, this is an ongoing process. This might be a long process. And my recommendation would be to go step by step, to start with one single trial, to perform a retrospective analysis, as Alexis explained before, to see, okay, we thought that we had 30% of waste at the beginning of the trial. That's what we planned for. But then I remembered that we had to add an extra packaging release. The recruitment was delayed. What was the real level of waste at the end? And then you try to learn from it. Where did most of the waste occur? Was it because we kept a lot of a large safety stock at the depot? Because we loaded the sites with buffer levels that were maybe set too high? All the sites were set to high recruiter, even though they were not recruiting that fast. So you try to learn from the past. And from there, you apply it to one new trial, then a second one, then you try to influence the processes. You discuss with your colleagues from the clinical supply chain team, from clinical operation, and you see, okay, what can we do? Implementing this process gives visibility on the real patient demands. So it's you no longer have to rely on best guesses on Excel sheets to make sure that you plan enough kits for your patients. It ensures that to lower the unnecessary waste and risk. It reduces the workload. That's very important. Reduces stress level for clinical supply chain manager, because you no longer have to deal with urgent shipments because there is a stock out in Brazil. If you can adapt the number of SKUs to those levels, for example, it's easier to deal with a trial with 4 SKUs, 4 different kit types rather than 12. If you plan the kit design in a smart way. And then something maybe for the next webinar, but let me know if that's something that that could be of interest for you. Reducing the waste can accelerate clinical timelines. So we often say that time is money, but money is also time. Because if you reduce the cost of the trial, you can reinject that budget into something else, into, I don't know, opening more sites, initiating new trials. So overall, it can increase your clinical R&D. So that's pretty much what we wanted to discuss today. And I think we're pretty much on time. We have about 15 minutes for questions. Thanks a lot for your attention. And yeah, if you have any questions, please shoot.
Yeah, we've already gotten a few really great questions in the chat. First, just like to say, Antoine and Alexis, thank you so much for doing this for everyone. It was a fabulous presentation. Got a lot of really positive comments in the chat as well. Thank you for your time. Thank you to everyone who's joined today. We appreciate you being here and for joining and for submitting some questions in the chat and participating in the poll. So I'll just read through a few here. First question, actually, just a simple answer that I can provide. Could you please share these slides? Yes, we could provide a recording or the deck itself after the webinar has concluded. Feel free to contact us directly, or we'll be reaching out to you individually as well once the seminar has concluded. So Antoine and Alexis, I'm just going to go through a few different questions here and answer to the best of your ability if possible. We'd love to get your responses here to a few questions. So first I have, I think one of the biggest areas of waste is supply sent to sites that don't enroll patients or well under enroll. I wonder if you've seen this in your data. Am I right or am I wrong?
You're completely right, indeed. Patients that do not enroll any patients still requires buffers. Those buffers expire, those kits expire. We have expiry events, so we need to send fresher kits. And that's precisely why we want to push for what we call the smart prediction. That's the case study that Alexis showcased just before. If you manage to have a minimum screening period that is longer than your shipping lead times, you don't need buffers. You don't need buffers because you will only send kits when patients will be in screening. So if no one ever shows up, you don't need any kits at this specific site. And another piece of advice that I would give you is to analyze the data like every 3 months, every 6 months, something like this, to see, okay, this site, should we close these sites? And it's a tough question, I know, but at some point, if you're in shortage of, I don't know, the comparator that you're relying on, that's a question that is worth asking.
Great. Thanks so much, Antoine. Another question here. In master protocol designs, for example, basket trials or umbrella trials, how much or how long can we reduce safety or waste stock?
That's very interesting because we've been working a lot on those types of trials recently. The answer is that it depends. It depends on, I cannot give you one single answer because it really depends on the design of your trial. How many SKUs do you have? What type of shelf life are you working with? If you're working with 6 months or with 48 months of shelf life, it's going to be a totally different answer. So it depends also if the trial is global or local, how many patients per site they have. So to me, the best is to test it out in the software to assess, okay, what's going to be the impact of my baseline? What's going to be the KPIs of my baseline? And then what if I change a couple of parameters? What if I reduce the safety stock? Does it generate some risk or not? Can I tweak the IRT parameters? How often should I resupply the sites? And that's an optimization that you can run. And yeah, honestly, without seeing the trial, it's very difficult to give an answer. And there was another question, I believe. What kind of overage do you need for such type of trial? Again, it depends. You've seen in the slide before with the release frequency, the shelf life has a significant impact on the type of overage that you need. And that's really a question we get asked a lot. What kind of overage do you typically see in oncological trials? It depends. Okay, it depends. You need to test.
Great. Another question here. Do you have any experience with trial designs that have very few face-to-face visits where IMP is assigned? For example, 2 face-to-face visits with a visit frequency of at least 6 months. What are your recommendations for waste reduction?
Yeah, very interesting. For this one, what I would suggest is, first of all, it depends again on the shelf life and the number of patient per site, consideration like this. But I would advise you to have a flexible supply chain in the sense that to have multiple packaging runs. Because if you only plan for 1 or 2 packaging runs, then you plan already for a very long time and you don't know what's going to be the state of your trial in 6 months. So in 6 months, things might be very different. It depends on the dropout, it depends on the indication, but I would advise you to have flexible supply strategy.
Great. A few more questions here. Can this trial design be done on every trial?
Yes, to a certain extent, yes. I would say that we cannot ensure such the same level of waste reduction for every type of trial. Let's face it, some trials are inherently with a very low waste level, and that's very good. Some trials are extremely wasteful. Some designs, some types of indication are extremely wasteful. If you have, I don't know, 8 kit types multiplied by 3 different booklets, then you need a lot of stock at the different depots. Then it starts to be messy and to generate a lot of waste. In the same way, if you have very low probability of titrations, but you still need to keep buffers at the site to ensure potential up or down titrations, then again, those buffers have a very low chance of being consumed, but you still need them. So it depends on the type of trial, but overall, yes, this could be applied to any type of trial.
Great. How can you influence drug substance overage? From my experience, drug substance is manufactured long time before study protocol preparation or country selection.
Yeah, that's a fair point indeed. What we tend to do is to consider future demand. So we discuss with the clinical team, with manufacturing, see, okay, how many trials do we expect in the next 3-5 years? We plan for a high-level forecast, of course, not as detailed as the one that you've seen before, but something high-level. And then again, we try to be flexible. If possible, we try to have different batch sizes, because at the beginning of the program, maybe you don't need a batch of 10 kilograms. You may be able to work with only a batch of 1 kilogram. Depends, of course, of the fixed cost, on the availability of the CMO, on the manufacturers. But we tend to push for more flexibility so that you can react to unexpected events. Unexpected events being a different yield than the one you expected, or Clinops wanting to initiate two new trials, or to increase the number of cohorts, number of patients in this specific trial, so that you have some time to react. But indeed, as the drug substance is manufactured long before the trial starts, you need to plan for those uncertainties.
Great. Another question here. How do you convince Clinops to follow your recommendations?
That's a good one. We don't want to convince them. We want to include them in the process. So that's a different kind of mentality. I think if we're being honest here, I think we need to start with data. So if you tell Clinops without any kind of data, I believe we should have a different screening period. I believe we should have a different visit schedule. They will just say no. And that's completely understandable, because it does not go in the direction of more patient centricity. While on the other hand, if you go to Clinops, if you discuss with them and you say, what if we would change that? Look, it would save us 3 million, and it would significantly reduce the waste. It would mean fewer shipments for my team. That would reduce the workload. So is that something that we could consider? Does it really have an impact on patients? And if that's the case, if it does have an impact on patients, let's just weight the pros and cons. Because again, sometimes if you see that you're saving 20 million by adding a controlled visit so that you can lower the buffers at site level, maybe it's worth doing it. And again, it's an informed decision, but at least it's not a gut feeling of just pushing back.
Makes sense. Just, I think, a few more questions here. We're almost at the end of time. Could this all be achieved in Excel?
Oof. Short answer is no. And to me, there are two main aspects, two main reasons why this cannot be achieved in Excel. The first one is that in Excel, you're still relying on the manual overage. So you have to enter how many, what kind of waste level do you want? Do you want 20%, 60%, 100%? And it's not a tool that is looking at all the moving pieces at the same time and giving you the right answer. So that's the first thing. And on the second aspect is that Excel does not consider the level of risk, the risk of misdispensing and the risk of drug shortages. That's definitely a no-go in clinical supply chain. We don't want to be a bottleneck. We don't want to have to pause a trial because of supply issues, definitely. So if you rely on Excel, what we observe in the industry is that most people overestimate the demand, overestimate the quantities to pack to make sure they never end up in such a situation. But if you can combine the two, low waste while ensuring no risk, that's a win-win.
Great. And a lot more questions here coming in chat, but I'll only pick a few more as we're running out of time. Could you explain more the part on retrospective analysis and the link to waste? I believe we manage our trials with 30% priority to FPI.
Yeah, and if you have 30% prior to FPI, that's great, because that means that the indication that you're working on, the number of patients that you have in your trial is okay with such a level of waste. Sometimes we start with 60%, sometimes with 10%, it really depends. What is important to consider is how do you define the waste? I explained that quickly before, but sometimes when we discuss with clinical supply chain managers, they say, oh, we have a very low level of waste in our trials. And then when you dig a little, you start to understand that their definition of waste is just what they add on top of everything else, on top of the safety stocks, on top of the of buffers. So we first need to clarify the definition of waste, and then if you start with 30% of waste at the beginning of the trial, that's your intention, that's what you expect, then life happens. Then you have a delay in your recruitment. Then Clinops, I don't know, if you have an adaptive trial, wants to open new cohorts. I don't know, there is a shortage in this specific comparator, so you have to do some manual action. And overall, if you're not optimizing your trial, you will end up with a waste that is generally higher at the end than at the beginning. So that's why it's very interesting to have those retrospective analyses to see, okay, how much did we really consume and where did the waste come from? Did it come from the sites, from the safety stock at the central depot, the local depots, and what's the reason behind that waste?
Got it. Great. And last question here for you guys. If we ship less kits in order to avoid waste, we will automatically have a higher possibility to trigger even more shipments. Each shipment has a cost. It would be interesting to put these costs under consideration, which tend to balance the cost saving of avoiding wastes, don't you think?
Fully agree. Yeah, that's something we already do. It's part of our standard process. We have what we call a standard optimization curve, where we have two axes, the cost and the risk, and we vary the frequency of the shipments and the buffer levels. And what we do, indeed, if you're only shipping very cheap drugs, then it's not worth sending frequently. It's worth having a bit more waste and saving in shipping costs. While on the other hand, if you have very expensive comparators, it's worth reducing waste as much as possible, shipping very frequently. It's more workload, it's more shipments, but overall, in terms of pure costs, it will be more interesting to ship frequently. So there is always a trade-off. I fully agree with you.
That's great. So first, I'd like to apologize if it didn't get to anyone's questions or comments. We're happy to answer individually after this call. Again, we're also happy to set up one-on-one meetings, answer any of your questions individually, as well as book a meeting directly either with myself or with the solution engineering team in order to do a protocol and assumption analysis complementary. We thank you all for joining our call today, and it was a pleasure speaking with you and teaching a little bit about designing a clinical trial. Antoine or Alexis, any final remarks?
No, really don't hesitate to reach out if you have any question, if you like the slides, if you want to discuss that further, we'd be happy to do so.
Great. Thank you all very much.
Thank you all. Bye-bye.